Okay, so a very warm welcome, Christian, to our EvoKE webinar. We're very glad to have you here as a speaker. Actually, so first of all, to introduce you, I think I should mention that your last name, Klug, is actually German for smart. So, and I think that like describes you as a person probably very well, since you're a smart person. You're a
Thanks.
paleontologist. You did your master's degree and your PhD at the University of Tübingen in Germany, and then you moved countries to Switzerland, just like me. But you did your habilitation, and now you're titular professor. I had to Google that, what that meant, but you're a professor now at the University of Zurich. You dedicate, and your research is dedicated to the paleobiology, especially of the Paleozoic and Mesozoic era, and you study cephalopods and early vertebrates. And we got to know each other when I had a talk at your institute, and I tried to tell everyone in the audience how sad it is that we have so like not much interest in plants. And then afterwards you approached me and you said, yeah, it's the same for me when you talk about invertebrates. Everyone wants to hear about the vertebrates, and not only that everyone is interested in animals versus plants, but also like in mostly invertebrates and not so much in invertebrates. So I hope that that today you can you can uh, how would you say that you can
Convince.
convince us. Thank you. That was the word that I was searching for. Now I hope that you can convince us and show us how interesting invertebrates really are. And so I give the stage to you. And yeah, thank you very much again for being here.
Thanks a lot for the invitation. I hope I won't disappoint. And first, I guess I have to share my screen. Let's see. I haven't done it in a while. Share. Hold on. I probably have to first go in display mode. No, this doesn't work. Up. First, I have to share screens here. Share screen there. Um, this one. Share. So does it look good to you? Do you see
That
my
looks,
slides?
we now see the presentation mode.
Ah,
So
okay. So I
can
have to try a
you,
different one.
mhm.
Uh,
But while you're searching, I will just tell a little bit more because I forgot to mention that you also are the curator of the
Now it's
museum, the
better.
Natural History Museum in Zurich, and and that you
Is
also
is
teach at the University of Zurich and the ETH.
is good now?
Now it's good.
Okay,
Perfect.
hooray, hooray. So we can start. Okay, so welcome everybody. And I suggest if there's questions, if I'm too fast, if something is not clear, if you're curious about something, just interrupt me, okay? And so this is kind of a more open dialogue thing than a blunt, boring monologue, I hope, at least. So let's see how this works. Yeah, now it works. So this is what I'm going to talk about. So anatomy of cephalopods, anatomy of ammonites. Then I have a bunch of questions because these questions often arise when I talk to people about ammonites. So how many arms did they have? Did they have a hood? Did they have an ink sac? Did they have lens eyes or camera lucida eyes? How many gills did they have? That's relevant for their evolution and phylogeny. Why did they become extinct while Nautilus survived? And I think I have a good answer there. And one thing which I find interesting and amazing is the topic, how did ammonoids swim? Okay, so let's go. So first of all, here you see a very old illustration from 1832 by Richard Owen, the founder of the Natural History Museum in London. If you haven't been, you should go because it's a really beautiful museum. And what you see there quite nicely is that these nautiluses, but also ammonites and actually belemnites and even sepias, they have a chambered shell. Yeah. And this chambered shell is also called phragmocone and consists of many chambers that are separated by septa, which you can see very nicely in this drawing. And then these chapters are connected by the siphuncle. And the siphuncle is actually contains a living organ, and via the siphuncle, the animal is capable of exchanging water in the chambers by gas. And this is an osmotic process, meaning it is also quite a slow process. Yeah? And inside the fragma cone, there's actually environmental pressure. So there's surface pressure. So it's 1 atmosphere. And that is even more amazing because if you know that these animals live happily in depths of up to 700 meters, and then it's really crazy. Actually, imagine you have a shell that's kind of 2 or 3 millimeters of limestone, basically, and protects this animal with the surface pressure. Then just imagine you're in a submarine which has a shell thickness of a few millimeters, and you have this enormous pressure of this great water depth. This is just it. This always freaks me out that this actually works. If they go deeper, if they go below 800 meters, the shell actually implodes. Yeah, so it wouldn't survive that. And the front part of the conch is basically the body chamber, which houses most of the soft parts. And there you see nicely the arm crown. In this case, since it's the Nautilus, you see the hood, then the pinhole camera eye, and at the bottom you see the funnel. And the funnel, which is also called hyponome, is important for locomotion. Okay, so
Sorry
these
to
are some,
interrupt
yes.
you, but like, so can you tell us more about like how that works, that it
Ah,
doesn't implode or?
so the shell is actually consists of aragonite crystals that is embedded in a matrix of organics. So this is kind of a compound material like carbon fiber today in race cars and fancy bikes. And it's a super stable material which is combines the strong properties of both materials. And this is how this works, right? So if you would build a submarine, you would just take like, I don't know, 10 centimeters of steel and you would be happy.
Mhm.
And Nautilus does it the fancy way with aragonite crystals and organic materials.
So that is probably not something that we as humans like could imitate or that we could use.
It's a good question. It would be an interesting thing. Then we could maybe take some millionaires and let them try the submarine.
Okay. That's a good
Anyway,
plan.
so to move on, here is some yeah, let's say early forms of three important groups of cephalopods. On the top number under A, you see a black tyrannosaurid. These are the earliest cephalopods we know, at least. And the interesting thing is that they have a bunch of characters that are reminiscent of their ancestors, monoplacophorans, which is a weird group of mollusks that still lives today, but it is very, let's say, obscure because it lives in great depths and it actually was unknown from the living organisms for a long time. I was only discovered, I think, in the 1960s by a deep sea expedition where they found for the first time living representatives of the monoplacophorans. The monoplacophorans, they basically look like limpets, only that in contrast to limpets, they have serially arranged muscles and also serially arranged kidney-like structures and gills and so on. Yeah. And this is something which you see in A. So there's a series of muscles that actually can withdraw the soft body. And there's actually also several pairs of gills. That is hypothetical, I must admit, because we don't have soft tissue preservation of these half a billion year old animals. And the rest is probably quite simple digestive tract, simple jaws maybe, and for the arm crown, we also don't know because it's almost never fossilized. Then the orthocerids in the middle under B, they're important because they're actually ancestral to probably to all living cephalopods, so to the crown group of cephalopods, so all squids, octopuses, um, and also maybe the nautiluses. Yeah. So it's basically the same as an A, only they maybe already had less skills, possibly, but otherwise it's quite similar, just a bit more straight. The siphuncle is in the middle, so this is something that's different. And from this group, actually both the coleoids, so squids and octopuses, and the ammonoids, about which I'm going to talk today, derive. And one important step which we can actually track in the fossil record is that the siphuncle, so this little tube that connects all the chambers, was displaced from the center to the venter. Yeah? You see this in C, that this little tube is actually on the outside of the conch. Yeah? And in ammonoids, what also changes is that this conch starts coiling. And I will come to this, why this is relevant at a later point. And what's also interesting, maybe in these drawings, you see how the conch is actually very elongate, and also the soft parts become really elongate. And also, and this is something in which I managed to prove actually in 21, that ammonites really had only two gills. That's why they are called dibranchiates. Like modern squids and octopus, they also have only two gills, while Nautilus is a bit more ancient because it has four gills. Yeah, so it's a tetrabranchiate form. Okay, so these are these early forms, and then we go into soft tissue preservation. So that is something that is, of course, very rare because, as you can imagine, in these very old fossils, there are sometimes hundreds of millions of years old. Usually, what falls apart first is the soft tissues, and only the mineralized parts, so the shell or the jaws. This is what remains. So, for example, if I die in some time in the future, and I'm rotting away somewhere in my grave, at first all my soft tissues go away, my muscles, my heart, my lungs, and all these things, they rot. And then at the end, there's my bones and my teeth because they're mineralized. They consist of calcium phosphate. And actually, at the very last, my teeth would go, or possibly even stay, depending on where I die. Yeah. So, and to find soft tissues, especially in mollusks, so in all these animals that have actually a lot of soft tissue, not so much skeleton like we do, this is really exceptional. In ammonites, it's even more exceptional because imagine if the conch is preserved and there might be the soft tissues inside, but you don't see them because there's the shell still around it. Yeah. So that's a bit of a problem. So in these fossils here, you just see some black spots and it doesn't look like much. And I must be honest, it took me years to understand these fossils. In the beginning, I wrongly interpreted them. And then after some discussions with various colleagues and so on, I went to a meeting and presented them as squids, but then I actually got convinced that they're actually ammonite remains. Um, and then I really started to understand all the anatomy. And then sometimes like this, you start understanding one part, and then you think, aha, if this is that, then next to it there must be this organ, right? So it's kind of a detective thing where you kind of figure out what is what step by step. And these fossils are one of the very few where we have parts of the digestive tract. You see crop and stomach is there, the jaws, and also the radula. I don't know if you ever heard of the radula. The radula is actually present also in snails. And sometimes if you have, I have to close the window because it's a bit noisy outside. Um, Regular, you see the traces actually if you're, let's say, in the park and there's a surface that is covered by algae or lichen, and sometimes you see these zigzag patterns that are produced by snails. And then they rasp off the algae with these tiny teeth which they have on their basically on their tongue. It's a rasping tongue. Yeah. And if you have them in a filthy aquarium sitting on the aquarium window glass, sometimes you can actually see them if it's a fairly big snail and you look what it's doing, then you can see actually traces of this radula. And ammonites also had a radula, and in this case, it is preserved. You see it in A, B, and E actually, and in A and B, you see that there's one tooth sticking out at the top, and this is very typical for the baculitids. So it's one group of Cretaceous ammonites, and we know this from other better preserved specimens actually. So this actually helped me to become sure that it's really a baculitid ammonoid and not a and not a squid or so. So in E, you even see something that might be the arm crown because so as you probably know, the cephalopods are the most intelligent invertebrates. They have the largest brain, and their brain is actually like a little bit in our case is located around their throat, around their esophagus. So to protect it against any hard art particles that might be ingested is actually in a capsule, which is made of something like chitin. Yeah? And this actually sometimes preserves, and I will probably show you some more of these structures. So, and this is how you sometimes can identify the head and the eye capsules because they're also um, in chitin. And what you see in E, these dark blobs where it says cephalic cartilage, this might actually be the eye capsules. And in front of the head, there's not much else but the arms. So I think this fuzzy thing on the top where it says arms with a question mark, this might be one of the very, very few cases where arm remains are preserved. But yeah, it's not much, I know. But that's the best I can offer in this context. So,
But can you tell us, like, so what are the conditions that have to be, like,
yeah,
what were
yeah,
the
yeah.
conditions of this fossil that this actually preserved?
Yes. So, of course, it has to be embedded quickly. And often this is in layers where at the seafloor there were low oxygen contents. Yeah. Often these are actually dark sediments. In this case, it's not very dark, but the obscure thing is that sometimes if you have these dark sediments and then they weather, they lose their organics. So what might have been dark at one point might have lost the organic content and then become actually light colored. So that's kind of contradictory, but that's what's happening sometimes. And this is actually from some sort of black shale layer. So it was probably from a basin where the conditions were sort of anoxic, so there were no scavengers, and also the decay by bacteria would be slowed down, and then it had to be embedded fairly quickly, and then depending on the physiology of the organs, it could be either phosphatized or carbonized, so preserved as pure carbon, basically. And sometimes, well, alternatively, it can be pyritized. So these are three processes that actually usually helped to preserve soft tissues preservation in pyrite, so iron sulfide, or apatite, which is calcium phosphate, or in carbon. Yeah. So these are three pathways basically. Okay, let's see. Yeah, so here is another case on the left from Greenland where there's some soft tissues preserved. So in the back, the black spot is probably the stomach. In the middle, there's something that might be the gills, and in the front, there's the jaws. The jaws are depending on the group. They can be actually calcitic in Jurassic and Cretaceous forms, but in older forms, they're usually made of some sort of chitin, which is under certain conditions preservable. Yeah. But what's important for ammonites is they mostly have coiled conchs. And on the right, this golden thing, there actually the entire conch is preserved in pyrite. It's from the Hunsrück slate. And this is one of the most primitive ammonites. So it is one of the simplest forms where you see it's actually just kind of began to coil evolutionarily. And it already formed one whorl, but not much more. So that's kind of a cool specimen because this is what you can imagine a very, very early ammonite to look like.
So it's like a missing link. Could you say so?
Mhm. So it's a bit more complicated because both squids and octopuses and ammonites derive from the bactritids. And so therefore the bactritids are actually a paraphyletic group. And technically ammonites and squids and octopuses belong to the same group and should, depending on what you look at, how you look at it, be called either coleoids or they should all be called bactritids. Yeah. So that's a matter of debate if you want. So in that sense, it's a transitional form and not really a missing link because it's sort of within a larger monophylum. Yeah? But it's a transition from those forms that have straight conchs to those that have coiled conchs. As you see here in this slide, you see when they're more derived, they're really fully coiled. And that's actually interesting because it goes with an increase of complexity of the septa. And the septa, this is what actually makes ammonites appealing and interesting. And that's why they're actually also sometimes quite beautiful, I find, because these intricate patterns, they sometimes people ask, oh, are they plants? And I say, no, 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 that's part of the conch, actually. It's the septa, and they're actually, well, in this case, like in F, G, actually the shell has been polished off. In C and D, the shell had been polished off, but in this case, the shell is preserved in calcium phosphate. So it was acid edged out of a limestone concretion. That's why you have the freestanding septa there. Yeah. So it's quite unusual, but it's really cool to see the spatial arrangement of the septa in this specimen. Yeah. And what's interesting is we have an increase of this folding, of the frilling of these suture lines through evolution. So they start off like in A with very simply curved septa, and then gets a bit more complicated. You see this in B, where it makes this little hook. And then it gets kind of crazy. In C and D, you see that the parts pointing inward, they have this little frilling, these little hooklets. And then in E, F, and G, you see that all parts of the suture lines are heavily folded. Yeah. So there's a strong increase in complexity and folding, but this applies only to the outside of each septum, because if you look in H or E, you see how in the center of the septum is actually just slightly gently folded, and only towards the outside this frilling occurs. Yeah. This is kind of interesting, and we'll come back to this in a second to explain, because since a couple of years, we actually probably know what this means. So, and this is also interesting because through ontogeny, so through the development of one animal, this kind of recapitulates this evolutionary process from simple to complex. So in A, you have this transition from the first suture lines, from the baby, from the hatchling, so to say, or from actually inside the egg, where it's very simple. The suture line is very simple, getting more and more complex with each new wall that's being built. Yeah. So you see how this increases, and in B, you see actually the septa, so these chamber walls. Looking at these chamber walls, in black would be the lobes, and in white would be um, the saddles you can see there. So you see how this becomes more and more complex, and there's a bunch of drivers. So interestingly, it's controlled by embryo size, because the smaller the embryo, the more septa they build, and the more complex it gets with each step they make a new septum. Then with the whorl section, you see this also very nicely in B, because when you have narrow parts where you have the overlapping whorls, in these narrow parts, you get extra little lobules, extra little folds in the septa, and also depends, of course, on the growth rates of the septal mantle and of the opening of the conch when it grows, when it gets bigger, right? So there's a couple of controlling factors that actually um, help us explain the complexity of some of these beautiful septa. And here is one of the most important driving factors, because intuitively one might say, ah, yeah, obviously the septa, they're made to make the conch basically more stable so it can withstand the hydrostatic pressure from when they dive deep, the pressure from the water on the gas-filled spaces inside, because it has just one atmosphere. So it needs something to make it more resistant, right? Um, but those animals today among the cephalopods that go the deepest have actually the simplest septa. Nautilus has simple septa, and Spirula has very simple septa. Yeah? So this doesn't work. So Robert Lemanis and Igor Slotnikov, they had the clever idea. They built digital models, virtual models, and then they used a technique that is called FEA, finite element analysis. That's something that comes from the car industry, where they actually assess the mechanical properties of car parts. And so they basically, like here, um, they make a virtual model of the car part, and they put some artificial virtual stress and strain on these car parts to understand where is there some mechanical stress, which parts have to be reinforced, where can we maybe save material, right? So this is where this method comes from. And what they found out when they applied this to different kinds of septa, so thus such which are strongly folded and such which are not so much folded, is that actually it increases the resistance against point loads. This means, but maybe you can guess this already, this actually is probably anti-predatory, because point loads you get when something tries to penetrate your conch with the teeth, right? So that's probably the main driver, and that's how this got so complicated. And there's actually a bunch of co-evolutionary processes between vertebrates, especially fish, and the cephalopods on the other side. So this is kind of a driving each other. Yeah. Okay. So where do they come from? What's their origin? They come from bactritids. You see, actually, where you have this red star, this is the origin of the bactritids. And the bactritids actually just originate just shortly before the ammonites, a couple of million years earlier. Um, and they have, most of them have straight conchs with a ventral siphuncle. They're very simple, small, unsuspicious looking fossils, and most people would probably not even bother to pick them up. And but they're important because they gave rise to all ammonoids. And as you can see, the colloids, this comprises squids, octopuses, cuttlefish, and so on. Huh? So that's why the bacterids are important. They became extinct already at the great mass extinction at the end of the Permian, where 90% of all marine life actually became extinct. And that's where we lost them. And they gave rise very early to ammonoids, and actually very nicely can see the transitions. And the main character, and this is kind of funny, of ammonoids is actually the coiled conch. That's it. There's not much more than that because actually, if you look at the earliest ammonoids, they're just a bit more coiled than the bactritids, and that's it. There's no other difference. So, what's amazing though, we have a lot of fossils giving us a lot of information about what's going on, about this transition, also in the early evolution of ammonites. And what I find amazing, what I did with my first PhD student, Kenneth DeBartz, like 10, 15 years ago, um, we looked at the embryos. Now you might wonder, how do you know about the embryos? It's very easy. Since ammonoids grow by accretion, meaning they always, where the arms are looking out, they always add a little bit of limestone, a little bit of aragonite. Yeah, that's how they grow. But they always retain the embryonic shell in the very center of their conch. Yeah. So, and we also see where they hatched because when they hatched, they shortly stopped growing. And then, actually, if you imagine this might be the shell tube, they actually started in to add more shell in the middle. And this has, as a consequence, it has a little constriction there. So you see that it's kind of a little furrow on the fossil, if it's an internal mold at least. And that's where you can tell, aha, this is the point where the animal hatched. So that's how we know, aha, this is the hatchling shell. This is the ammonitella, as we call it. This is the embryonic conch. And that's now interesting because within the Devonian, so within the first 10, 20, 30, 40 million years of ammonoid evolution, the size of the hatchling decreased from, let's say, 6, 7 millimeters to about 1 millimeter. And the conch size increased at the same time. And then, as you see in the top here in this figure, you can also see that the volume of the body chamber, so the part that housed the soft parts, also increased quite dramatically. Yeah. So then we actually assumed a proportion of the volume of the body chamber for the ovaries and then made an estimate for the number of eggs they produced. And this is quite dramatic because we can assume that from the first ammonoids, which maybe produce something between 50 and a couple of hundred of eggs, the forms in the late Devonian, so a couple of tens of millions of years later, they already were in the range of hundreds of thousands of eggs. Yeah. So there was a drastic increase in fecundity. Yeah. So that's very interesting, I find. And if you look at the relation of egg size and the number of eggs produced, so in the vertical axis you have the number of eggs, horizontal axis is egg size. It's don't be confused. This is logarithmic, yeah. And there you see ammonites are really on the side with the lowest size and the greatest number of eggs. Yeah. So that's important to understand the ecology of these animals. So they're what was formerly called R strategists.
Sorry, and so now I understand where you found the embryo or how you can tell how the embryo looked like. Is that something, by the way, is that something that we as like lay people could also see if we looked at an ammonite, or is that something that you need to scan or
So it depends.
something like that?
Sometimes you can actually see it with a hand lens or so, or with a microscope, but it's really small because if you imagine the embryonic shell is like
Mhm.
a millimeter or so across, yeah,
Mhm.
then it's a really tiny structure, and
Okay.
you need really excellent preservation to see it.
Mhm.
And often this is actually covered by the outer world, so you don't see it. Or
Mhm.
there's still sediment covering it, so you need to be lucky to see it. But I think once you've seen it once, you can recognize it. It need a lot of, let's say, intellect to recognize it. Once you've seen it, it's actually you can recognize it in other fossils fairly easily. At least if you have good photos or a good microscope of a good with a
Mhm.
good specimen, it's not so difficult to see.
Okay. That's so
Okay.
interesting. And but the other question is, but so how do you know how many eggs an ammonoid had?
Well,
Ammonite.
oh, that's the next slide. We
Oh,
have a
sorry.
few, no worries. We have now a few specimens. This one doesn't look like much, but it's actually beautiful. The beautiful colors come from the original aragonite because, as I told you, the shell consists of alternating layers of aragonite and organics. This means that light is diffracted all the time, like in the mother of pearl, nacre, and this produced these beautiful colors. In this case, the original aragonite is preserved, which is actually rare because it easily deteriorates to calcite, and then these colors are gone. Um, but on top of that, this specimen has a lot of embryonic conchs inside its body chamber. Yeah, and you can see photos on the right side. Um, it's not that easy to understand, I think, but what you can see is each of these subcircular structures is basically a baby ammonite, and in E and F, you see there's these white structures, and these are actually the lower jaws of these babies. So we really know at the time when these animals died, these animals were complete and they were probably inside the body chamber of the mother animal. And then we can count and make a rough estimate of how many eggs, how many embryos there were. Huh? So this is one way, and the other way is the entirely estimated way, as I suggested. We make an estimate of what proportion the ovaries might have filled, and then we make an estimate of egg size based on the embryos, and then we can make an estimate of the number of eggs. That's what we did for this other graph, actually. All right. So, but it's good to have both because then we can test each other, and we can see, does it make sense? Are we completely off with either one, And that's very interesting. What's also interesting, we now know we can actually sex the ammonites, at least some. So we can determine the sex, and I will come to that in the next slides, actually. So already in the 60s, both a Polish colleague and a British colleague simultaneously in the same year realized that there's two forms of ammonites occurring in the same layers, especially in the Jurassic. They look from when they're adult, they look dramatically different. But if you look at the inner worlds, they look exactly the same and they always co-occur. Yeah. So this told them, maybe they're actually males and females. And back then they weren't sure what is what. So they called it microconch and macroconch. So the big conch and the small conch. The microconch has these strange lappets sometimes or strange aperture, strong as an adult, sometimes strong ornament. And the macroconch is usually larger, has a less strongly ornamented conch, and usually a simpler aperture. Now, um, intuitively, I always said probably the small ones are the males because there's some good examples from modern squids and octopuses where this is the case, where the guys are tiny and the girls are rather big. So the guys are basically serving just one purpose, transforming the sperms, and that's it. And while the females have the actual work to do, the actual workload, as so often. So yeah. So this is probably was the case in many ammonites as well. And this thing here, I actually got this before COVID. And then during COVID, I didn't have to do so much museum work. So I could actually look into my research a little bit. And I tried to solve this riddle. And I step by step managed to understand this fossil. So on the top left, you see the apticus, which is the lower jaw. This was obvious from the beginning. What's also obvious, there is no shell. Yeah, where's the shell? But that's something I will come to in a second. I think actually I have too many slides, so I will stop somewhere in between. Isabel, you tell me when it's enough because I have way too many slides. Um,
If you when you sit here alone in the Zoom room, then you know that you went overboard with the
No,
time.
yeah, yeah. Actually, I have an appointment later, so I have to stop at one point.
Okay.
But it doesn't matter. Okay, I just keep on talking, and then at one point we call it a day. In the worst case, I can give you another seminar and present the other slides to you if you want. Anyway, so there's the apticus, the lower jaw, which is mineralized in many Jurassic forms, and then um, yes, and then I also, which I didn't do and which I don't show here, is I also took UV photos, and in ultraviolet violet light, um, phosphatized structures start to shine. And in this case, I this helped me to identify the esophagus, and also, there's another thing which became clear because if you look at the top right, you see there's an elongate structure, and there's something which looks a bit like, yeah, well, it actually does look a little bit like shit, and this is actually shit. So this is the intestine of this animal, and this sausage-like reddish thing that kind of curves on the right side. This is probably the stomach. So I managed to homologize bit by bit there. Yeah. And and then you see on the right, you see these kind of irregular paired structure that points kind of downward. This is the gills actually, and that's important because this shows us they had only two gills, and they're hence this underlines our interpretation that they were closely related to squids and octopuses because they also have only two gills, while nautiluses have four gills. Yeah. So that's kind of interesting. And then I also looked at the structure, which is kind of central. It's very difficult to photograph because the structures are super flat and there's not much contrast. And I identified those actually as the male reproductive organs, which told me that this was actually a guy. Yeah. So I was able to sex it. Here you see photos of it. It's really hard to see. But the interesting thing is it had the same proportions and structures as the reproductive organs in Nautilus. So you see this in the bottom, there's a drawing of the Nautilus organs, and then I colored it the same way, very suggestively, in the fossils. And I'm pretty sure these are really the male reproductive organs. Yeah. And what's also interesting now, the question, why is there no conch? So if we look in the fossil record, we often find ammonites that miss a little bit of the shell in the back of the body chamber. And there was an earlier paper by Adia Klompmaker already, yeah, almost 20 years ago. And back then I thought, this is bullshit. I didn't believe it, but now I believe it in the meantime because there is a pattern. It's always in the back of the body chamber. Now, if you understand the anatomy of ammonites, you know that the main muscle actually sits in the back of the body chamber, is attached to the shell there. So if you want to get the animal, if you want to eat the animal, if you come from the front, the animal just withdraws its arms into the shell and it's hard to access. But if you go to the back, you break the shell open, you basically rip out the muscles, then you get all the soft parts, all the goodies, yeah, and nothing of the shell. You basically have only the soft parts, and this is how I reconstructed this. Yeah. Now, who did this? Many layers where we find this, there's no fishes or hardly any fishes, but there's a lot of octopus relatives, early octopus relatives, and octopuses are clever, so they probably learned if I crack the shell this way, I actually get hold of the entire soft parts. And we actually have fossils of fossil octopus ancestors that have actually the lower jaws of ammonites in their stomachs. So they probably really did this. Ah, yeah. And there's another specimen which I got hold of 2 years ago. It's from a collection in Chicago, and this is the counterpart. So this is actually a female. You see again, what looks like a bivalve is the lower jaw. And you see on the left side, you see white light photos. On the right side, you see a UV light photos, and then my interpretations as drawings below. And this is quite amazing because this is from Solnhofen, one of the famous conservation deposits where you have exceptional preservation. And in the very back of the body chamber, you see all sorts of remains of soft organs. And here is a close-up or some close-ups And this is probably the ovaries that are preserved there. Yeah. And you can even see vaguely more or less circular and spiral structures. And these are very likely the embryos inside the eggs that are preserved there. And this allowed me to make another estimate of the number of offspring. And this helped me to make this graph, which you see here, several genera with various estimates. And in this case, no, Neochaetoceras is that fossil that I'm showing here, um, where you have well, I indicate a range of the number of eggs somewhere in between 1,000 and maybe a couple of tens of thousands of eggs. Yeah? Paraputzosia, by the way, is the largest ammonite that has been found so far, which has a conch size of over 2 meters. And but still, the embryos are tiny. They're 1 millimeter, right? So there's plenty of space in a conch that is 2 meters in diameter. So we estimate that this one produced something in the range of tens to hundreds of millions of eggs. And if you know about biology, for example, among fishes, there's mola mola. I'm sure you know it, the sunfish, this really awkward looking fish. If you don't know it, Google it. It's an awesome animal. And it gets huge. It's the largest bony fish, which weighs up to 2.5 tons. And the babies are tiny and they produce actually up to 300 million eggs. So one mother is very productive. And this was probably also the case in ammonites. And this is how I reconstructed this animal. In B, you see the male with maybe, this is speculation, we don't have proofs for that, maybe a modified arm pair to transfer spermatophores. And then in A would be a female. And you see the ovary in greenish in the back of the body chamber. You see the big jaws, you see the eye, and you see the digestive tract and the one pair of gills and some other internal organs. Yeah? You also see the phragmocone with the complexly folded septa, and on the outside you have the siphuncle that connects the chambers. Isabel, I suggest I just talk till I don't know 5 to 4 or so, and then we make some questions and answers, and we call it a day. Is that a
Yes.
plan?
Yes, that sounds good. Just
Okay.
maybe present us now like your favorite parts, like the
Ah,
candy in your talk.
okay, so number of arms, this is something which is a widespread question, and actually there's not many fossils. There's a few fossils from one groups where we have arm hooks. They look really strange, and there's just one family where we find this, and this is probably a pair of modified arms, as you can see here. And then a question for the hood. There was some paper back in the day in the 60s actually, and this they thought they had fossils of a hood. So this what you see in the orange oval here in Nautilus, but this was just a shell fragment. So ammonites didn't have a hood. Did they have an ink sac? Sometimes you see that, and the problem is ink is basically melanin, which we have in the skin and in the hair. Um, and also in our livers, by the way. And that's a substance we find in many animals and in many organs. And so the same holds true for ammonites and sometimes even the shell. And so some people found melanin. They said, they must have had ink sacs. But this doesn't make sense because um, the ink sac evolved in early colloids with the internalization of the shell. The lens eyes, I would say probably yes, because all living relatives, meaning squids and octopuses, have lens eyes. And Nautilus is not as closely related and has a pinhole camera eye. So I favor the interpretation that they had lens eyes. Gills, we've talked about that. So the dibranchiates, why did they become extinct? That's also an interesting story. I told you already about tiny embryos, right? So really tiny, tiny. Nautilus has fairly big embryos. They're like 1 to 2 centimeters. So they're, in terms of volume, they're orders of magnitude larger, right? Now, in addition to that, Nautilus has much lower metabolism. Nautilus sometimes lives happily on the empty shells of the molds of crayfish. Yeah. So when they shed their skin, they basically eat the skin and they're happy with that. And then they don't eat for a week or so. And then they make it maybe get another crayfish skin and they're happy. Yeah, we can even show this with stable isotopes. My former PhD student Ammonoid did this. He could actually demonstrate using stable isotopes that this is the case. So ammonites needed more food than nautiluses, but had much smaller embryos. And probably when the big meteorite hit Earth 66 million years ago, probably the primary producers went down, and probably the availability of food was low. And for Nautilus, it didn't matter so much because they could live without food for a little bit, while the tiny ammonites they couldn't. They didn't have the reserves. They need a lot of energy in a short time, so they didn't survive that. Huh? So that's my or our best explanation for why ammonites and belemnites also, for them it's also true, why ammonites and belemnites became extinct and nautiluses actually survived. So, let's see, is that it or yeah. So we could either stop, or if you want, I have one more chapter about swimming in ammonites. So what do you want?
Since we already started with questions in between, maybe just like tell us another chapter really,
One more
really
chapter.
shortly. Yeah,
Okay. So
because we
that's
still
really
have 3 minutes until
alrighty,
50.
right. So this is very interesting because if you look at the conchs of ammonites, it's very well preserved. We have nice 3D specimens, so we know how thick is the shell. We know where there's shell, where there's no shell. We know where there was the soft body. Yeah, the soft parts probably had a density of 1, like water, like us, more or less. Yeah, the conch consists of calcium carbonate. This has a density of something 2.6 or so, and the gas, well, it's almost nothing, right? So we can actually reconstruct distribution of mass within this animal. And I did this back in the day with little models, and you can do this also virtually today. And then you can identify the centers of mass and the center of buoyancy. And these two centers, they have to be aligned above each other when they're at rest. Yeah. So, and this actually tells us the position of the ammonites in the water column. So this tells us where's the aperture facing. If you have a conical shell, like here in Devonobactritis on the very left, they were more or less vertical, right? With the opening facing downward. Yeah. For them, horizontal swimming was difficult. They would basically do this, they would be very slow. Yeah. But then you see with the increasing coiling, the aperture moved up, and thereby the hypernome, where they expelled the water jet to swim backwards, um, became more or less aligned with the center of mass. And as soon as the hypernome was in level with the center of mass, they actually became reasonable swimmers. Yeah. So coiling is linked to swimming. So it made them better swimmers. Also, the further the hypernome was away horizontally from the center of mass, the more efficient turning movements would be. Yeah. So, and then this is something very modern. It's not done by me, but by some brilliant, brilliant colleagues from the United States. They made ammonite robots. So they built these shells following actually actual shells, and they placed also the center of mass by putting some weight and the center of buoyancy in the right places. They put a little engine so they could produce a water jet, and then they filmed how they behaved. And this is very interesting because if you look at all the different ammonites, they look so different. There's like tightly coiled, loosely coiled, um, some look more like snails and so on. There's really crazy shapes. And it was always the question, what sense does this make? So they filmed these ammonites and how they behaved, and they could actually demonstrate that different conch shapes. So not only how tightly they're coiled, if you see the inner whorls or not, and if they're broad or narrow or so on, they all had these all had reasons, let's say. This all had advantages and trade-offs, So it had there were reasons. Some were able to accelerate well, some were more enduring, some were very maneuverable so that they could steer better. Yeah. So this there's these three factors, and then if you look at the ornament, if they had ribs or were smooth or something in between, the ribs also had an effect. Yeah. So they helped them they were probably the ribs helped to avoid the rocking movement when they would expel water, but they would also increase drag. So they would become slower. Yeah. So you see there is ear well, sometimes there's layers where you have like 10 or 20 different ammonite species. People wonder why, what ecological sense does it make to have so many different species in one place, in one layer, actually. And now, if you understand this, this makes more sense. So they really were specialized. They had certain swimming modes. Probably can also assume that somewhere maybe closer to the seafloor, somewhere more in midwater or closer to the water surface. So there were specializations, ecological specializations, and that is quite amazing, I find. And that's where Peterman and Rutterbusch did some amazing work. They spent years into that, and it's really cool. So I find this really interesting. And here is my summary. If I'm not reading it to you, but you can read it yourself if you want. And if all of you have questions, I'm happy to answer them.
Thank you so much, Christian.
You're welcome.
I'm searching for the applaud function. No,
It's
no, there it
okay.
is.
Thanks.
Thank you very much for this amazing talk. I learned so much about ammonoids. And yeah, they seem to be pretty cool. So I understand why you're so fascinated with them. Even though I'm also fascinated how someone can like in these small structures like find so much sense. It has to like you have to dedicate probably like a lot of time. So if there is questions from the people listening, we would be happy to answer them. I think I was already like I already got rid of my questions. And if you don't have any questions, so I would be very interested, or we would be very interested to learn from you how you learned about the webinar. Like, did you find us on social media? Do you know EvoKE from other things? And so where did you find us? And what are topics that are interesting to you so that when we invite new speakers, that we can also have that in mind just before you leave, just maybe that you really quickly report us in the chat so that we get to know you and your interests better. And then, like, if there's now no question, then I would like to just like ask another question because I think the only thing that I didn't really understand is why the ammonoids became extinct and the nautiloids didn't. So I think that was
Okay.
too quick for my brain.
All right. Sorry, sorry. So it's actually an unfortunate coincidence of the size of the hatchlings, tiny in ammonites, large in nautiluses, and the metabolism, which is high in ammonites and low in nautiluses. Yeah? So therefore, nautilus babies were much more resilient. Yeah? They had more reserves because they were larger, and they had a lower metabolism, so they just needed less food. So in times with low food availability, low prey availability, they would be happy, while ammonites weren't. And actually, ammonites almost became extinct several times. So there were, and it's interesting. Um, Actually, I can show you the next slide. Oops, up. No, wrong. This one. Here you see, well, this is just part of it, but no, it's not, it doesn't matter. So they became extinct at the end of the Franian almost, at the end of the Devonian almost, at the end of the Permian almost, at the end of the Triassic almost, and then they became really extinct by the end of the Cretaceous. But they were four times only one group survived. And then they quickly evolved a huge diversity again because they probably partially because they had this large fecundity and they're also probably fairly mobile, so they could kind of distribute quickly. And but their small hatchlings made it hard for them, and also the high metabolism made it hard for them when somehow the food webs collapsed because of low primary production.
And was it also the asteroid that at the end of the Cretaceous like led to the ammonoids dying out?
Yes.
Oh wow, I didn't
Yeah,
know
yeah, yeah,
that.
yeah, yeah.
Okay.
So it's belemnites, ammonites, dinosaurs. Well, the non-avian dinosaurs, to be correct, right? So
Yes,
because the
thank you.
the birds the birds survived, as you all know, and well, there's a bunch of other groups that didn't make it, but yeah. Any other questions or comments?
Yeah, well, a comment and a question. So first, thank you very much. I just learn a lot, and I'm quite of fascinating of the way you are studying this. Um, and thank you for that.
You're welcome.
And I have a question. I'm not sure if I understand it correctly, but do you have like an explanation why some of them have more of offspring than the others, or is the this just related with their body characteristics or?
Yeah. So first of all, we have, let's say, direct information only for very few. Yeah.
Mhm.
So for most of them, it's kind of an estimate because what we basically did is we looked at modern squids and octopuses, looked at the size of their ovaries and how much offspring they produced, and then we took these values and transferred it to the volume of the body chambers of ammonites. Yeah. So that's how we proceeded. But when we compared these values to the values we got from where actually embryos are preserved, showed us that we are not completely off. Yeah? So there seems to be some relationship, conch size, body chamber volume, and the number of offspring. So this is somehow linked. And it also makes sense because there were thousands of ammonite species, and we can expect that there was some ecological disparity. Yeah, so they had very different strategies, very different habitats, very different foods, and so on. And then it makes absolute sense that they also varied a lot in terms of reproduction. And what is really interesting about this, actually, what I find very remarkable, sometimes if you think of ammonites, you just think of the fossils, like you know something like that.
Mhm.
You've seen those, I'm sure. And they're usually decent sized. You can pick them up, have them in your hand, and they're fairly big, maybe even very big, depending on where you are in the museum, maybe. Um, but because they had so much offspring, what was actually really abundant were the babies, the millimeter sized ones. So they were ecologically super important. They were probably food for so many other organisms, for filter feeding fishes and I don't know what. Yeah. And that's something which is also, I find very important.
And just other question that just came to my mind because you talk about this recent study um, using robots and things like that. Do you feel that, or how do you expect that these new technologies that we have nowadays, or AI, also can help us know even more about this?
Good question. It's a very complex question with many levels. So, um, hmm. So, I mean, these robot studies, I really like them, but there's a lot of effort there. So, until they had built the robots, have had fit all the bits and pieces inside the little engines and placed the weights in the right spot and so on. There's a lot of they spent years and years, and then filming them. They spent weeks or so in swimming pools filming ammonite robots swimming around and so on. So there's a lot of time in there, and there of course, AI can help you maybe calculating how you construct your robot, but in the end, building this, the AI doesn't help you too much. I'm a bit so. I think AI is great for certain purposes, especially if you have, let's say, great amount of data and very complex things you want to calculate, then it's super great and so on. And but I think I encourage people to use it very critically for a couple of reasons. One reason is doing research is great. It's good for our brains. And I mean, you're also in social studies, right? So if people stop using their brains, they get stupid. And we don't want that. We want clever people because clever people are better people, right? And so in that sense, I'm kind of saying, yeah, I'm very skeptical. Also, so it takes away jobs and it still uses a lot of energy. And in times of climate change, this is a strong argument, I find. Yeah. And I mean, if they build all these stupid data centers in the US and they actually cut off some private households from energy because they need more electricity. And I'm like, this is really nuts. And I mean, of course, I know AI becomes more efficient, and there's AIs that don't use so much energy. Some use a lot of energy, depending on the models you use. And I kind of try to minimize the use. I enjoy writing, so I write my shit and I make my own mistakes. But I think that's good, and I think we have to maybe embrace our mistakes more and see it as character and part of us, and rather than a weakness compared to AI. So I also, I like doing my own paleo art. So most of the illustrations, well, the one here on this slide is not. This was from another artist here from Zurich, Lily. But many of the artworks I showed, I've done myself. And this is a lot of actually manual labor. Most of them are based on pencil drawings. And but I know a bunch of paleo artists and other scientific artists, let's say. And for them, it's really tough because now it has become so easy to do um, art with AI. Of course, it often sucks, but some of it looks quite amazing, admittedly. And it has always been difficult for these people to live from their art, and now it's becoming even tougher, right? So there's another thing where I'm very skeptical concerning AI.
So I think these are really good last words. So the next time my boss asks me why I did a certain mistake, I will just say, because I'm better than AI and that's my
Because you want to save
human.
energy, you want to save the planet. That's why.
Yeah. This is my advantage of being human, that I'm able to make mistakes and recognize them. Um, Okay. Hey, Christian, thank you so much again for your time.
My pleasure.
I would love to hear much more about your work and your research. But you did an amazing job to spark our curiosity for invertebrates and ammonoids specifically.
Hooray.
And then, yeah, and then have a great week. And yeah, hope to see you
You
again
too.
soon.
Me
Thank
too.
you.
Have a great day, everybody.
Thank you. Bye.
Bye bye.
Bye. Thank you.